Good afternoon, folks. I hope this finds everyone doing okay today. This is part 2 in my lecture series on animal nervous systems. If you have not yet watched part 1, you might want to go back and do that so you have some background for this. And if you were in my biology class, you should probably go on the Blackboard site and get a copy of my summary lecture notes for this, the handout that will include important vocabulary and concepts that will help you to follow along with this lesson. So, last time we talked about the fundamentals of animal nervous systems, a little bit about the different kinds of nervous systems that we find in various animals, nerve nets, nerve rings, and centralized nervous systems featuring cephalization. We talked about the evolution of nervous systems as an internal mechanism for regulating the bodies of animals. Talked a little bit about why some animals have a more fully developed central nervous system than others, including the very interesting evolutionary question of intelligence and why some species are more intelligent than others. And we did some basic comparisons of control systems that are chemical versus electrical in animals, looking at nervous systems versus endocrine systems and how they help to regulate animal bodies. So what I want to do with this lesson is to go over some basic considerations of how nervous systems are organized and how they work. A lot of this is going to be universal across the animal kingdom. There's not a lot of functional difference in how neurons send signals in you versus how it happens in a lizard. And we're going to find that many nervous systems, particularly invertebrates, have more in common than they differ. There are going to be some important things that differ from one group of organisms to another. I'll mention some of those differences along the way, but what I'm looking at here is broadly going to be the big picture things that animal nervous systems do and how they do. Now, functionally speaking, nervous systems have three basic activities that they do, three basic steps of their function even, you might say, because these are sequential activities. We have sensory input, we have information processing, and we have motor outputs. Those are the three stages of nervous response, as well as the three broad categories of things that nervous systems do. Now, the one I think that in a lot of ways is the most interesting to look at across species is the first one, sensory inputs. That is collecting data and sending it from various sensory receptors to the central processing centers. That is directing information from the peripheral nervous system, or PNS, to the central nervous system, or CNS. After all, the animal can't make any decisions about what to do unless it has information to work with on what it is doing. So that is fundamentally what sensory input is for. It's giving the organism information about its environments. Now, that is going to involve a variety of structures that are specialized for purposes of collecting data, a variety of sensory receptors. Now, some of my sensory receptors are going to be well-developed complex organs, such as the retina of the eye or such as the cochlea of the inner ear. Others of them are going to be simple nerve endings that are present in the epithelium or in the connective tissue just beneath it. Some of those nerve endings are encapsulated, forming a little sensory receptor with a capsule of connective tissue to protect it and increase its sensitivity. Sometimes it's just a free nerve ending that is responsive to environmental stimuli. Fundamentally, what a sensory receptor does, no matter what kind of receptor it is a thing called sensory transduction. Transduction. And sensory transduction is basically converting sensory stimuli into electrical impulses, taking stimulus energy, converting it into electrical energy to send a signal along a nerve fiber. Now, thing you need to know about this, not all animals detect the environment in the same way. Not all animals are equally good at all the ways of detecting the environment. There are a lot of different possible senses that animals can possess. You might recall one of the basic details about life is that life is sensitive to environmental stimuli. And that means that all living things, not just animals, but plants, bacteria, everything else, has some capacity to detect what is going on in the world around them. They have cells or structures that are sensitive to particular kinds of environmental input. Sensitivity to a particular kind of data is what we call a sense. There are no all-purpose sensory receptors, I should point out, that there are different kinds of receptors for different modalities of data. Senses mean a couple different things, by the way. Your senses, first off, tell you about location of stimulus. Where is the stimulus coming from? Am I hearing the sound from over there? Am I seeing the object right here? Am I feeling the pressure on my skin right there? That's location. It's telling us about intensity of stimulus. Did I just get tapped on the shoulder, or did I just get punched in the shoulder? It is telling us about frequency of stimulus. Am I getting that stimulus, or am I getting that stimulus? And it's telling us about modality of stimulus. That is, what kind of data is coming in. And this is, I think, the really cool thing, that sensory data can mean a lot of different things. There are a lot of different kinds of information your body can collect about both its internal and its external environment. Keep that in mind. When we think of sensory receptors, our natural inclination is to think about what they're telling us about the world around you. That's not all of our receptors, folks. You have a lot of sensory receptors that are telling you about your internal environment as well. Whenever you feel your stomach hurts, whenever you feel pressure in your chest from a heart attack, whenever you feel pain in your lungs from breathing super hard after running really fast, all of that is internal sensory data that your body is collecting. I should also point out, a very important thing to know, is that a lot of the information that your body collects, you are never consciously aware of. Some of the data that we collect will produce sensations. A sensation is a sensory stimulus that you become consciously aware of. It is something that you know is happening. You're aware of that thing. You see that object walking in front of you. You hear that noise. It's a stimulus that you are aware is happening. Not all stimuli create sensations. First off, some stimuli create sensations that your conscious brain is never aware of at all. They're handled by other parts of the brain aside from the parts that do the thinking, and therefore your cerebrum, the thinky part of your brain, that's a technical term, is never alerted to that data. For example, you have sensory receptors in your blood vessels that are detecting things like changes in blood pressure and oxygen saturation and the pH of your blood. These are all important pieces of data that your body has to track. And without that information, it can't manage its own homeostasis. Reduction in oxygen level and an increase in carbon dioxide level, for example, will stimulate your brainstem to say, hey, you better start breathing harder to pull in more oxygen and clear out carbon dioxide. Or make an adjustment in your blood pressure, or release buffers into your blood to lower its pH, or whatever other homeostatic regulation needs to happen. So you've got a lot of sensory data that your body is collecting that you never become consciously aware of at all. You don't need it to be consciously aware because other parts of your brain are handling that stuff in the background. There is also data that comes from senses that you are consciously aware of, but you still don't get a sensation from it. Your body does not equally respond to all incoming data. You are constantly getting a whole range of data from a whole range of senses about environmental conditions around you. Some of these data are things that you will become consciously aware of. They will produce a sensation. Some of these data your brain will discard. Part of the information processing step that comes next is going to be sorting through all the incoming data and saying, what do I need to be aware of? What do I need to think about? What can I afford to dump? Your brain has to prioritize processing power for things that are most immediate, things that are most important to be paying attention to. And that means that relatively unimportant data is discarded without processing. And therefore, your brain basically says, this isn't important. I'm not going to get a sensation from it. It's not something I need to be consciously aware of. Sometimes this is background information that you will nominally pay attention to, but you will not necessarily pay a lot of attention to. You have no doubt all noticed that I have a blue background behind me. That is something that you're seeing. It's right in front of your face. It's probably not all that important in terms of what we're dealing with right now. Presumably, you're paying more attention to my words, my face. This is the part of the lesson that's probably drawing most of your attention. You're getting sensations from the sound of my voice. At least I hope you are right now. You're probably not consciously aware of the texture of your socks. Now you are, right? Because I just drew your attention to it. And that's a lot of times what happens, that your body pays attention to a stimulus when that stimulus has done something to draw its attention, when something has been pointed out to you, when something has changed, when something in the environment is moving, when something is touching you. This is the kind of thing that our brains will say, I need to pay attention to that. But if a stimulus is relatively static, our brains will tend to process it and then say, I don't need to do anything about this, and discard it without getting a sensation. I have just drawn attention to the texture of your socks. So you're probably feeling it now. You're probably thinking about it now. Previously, it was a constant sensation. You were wearing your socks the whole time, assuming you're wearing socks while I tell you about all this. But you weren't necessarily going to notice that sensation because 30 seconds after you put them on, your brain wasn't worried about the texture of your socks anymore. It was a constant stimulus that does not require any particular response from you, and therefore the data from that stimulus was something your brain was discarding. It required no processing power. It didn't need to be done anything. But the moment I drew your attention to your socks, your brain says, oh, socks, something important about my socks, and then you started feeling their texture again. Now, because your brain makes associations between related things, you're probably also consciously aware of the texture of your shirt or your underwear or whatever else you happen to be wearing right now, because I pointed out one of your garments. And now your brain is devoting processing power to saying, do I need to be thinking about how my sweater feels? Because this is something that has been pointed out to you. So we have a lot of data coming in at any given time. We don't necessarily notice all the data that is coming in at any given time. Now, how about the question of your senses? How many senses do animals have? Now, the answer to that is certainly going to vary from one animal species to another, because not all animals have all the same senses. Snakes, for example, are deaf. Snakes do not have a sense of hearing. That is not a sense developed in that animal. You can talk to them, but they won't hear you. Snake charmer, you all know the classic idea of the snake charmer, the dude who plays the flute for the snake, and the snake gets up and sways back and forth. That snake is not responding to the music. That music is for you watching the performance. What the snake is responding to, and if you notice, the snake charmer, while he's playing his flute, will be tapping his foot, because the vibration is something the snake has a sense of, and therefore that is what the animal is responding to. There are animals that cannot see. You've all heard the phrase blind as a bat. That is completely untrue. Bats can actually see better than we can in the dark. But there are certainly animals that are literally blind. There's an animal called the blind fish. It's a fish that lives in underground rivers in certain caves in the central United States. If you go to Mammoth Cave in Kentucky, which you should, it's really cool, then there are blind fish that live in Echo River, the subterranean river inside the cave system, that don't have eyes at all. They have no visual sense, period. They've adapted in that way because an eye is an expensive organ to run, and when you're living in a completely dark environment, it's doing you no good whatsoever. It's actually more evolutionarily beneficial to not have them. By contrast, there are animals that have senses that you don't have. We have learned, for example, that birds have an electromagnetic sense. They have an ability to detect electromagnetic radiation. They're sensitive to the magnetic field of the planet itself. And this helps them to navigate when they are migrating. It is by following the Earth's magnetic field, that birds figure out how to go back to the same nesting grounds when they migrate every single year. We have learned that the platypus also has an electromagnetic sense where it detects electrical activity in the brain of surrounding animals. It helps the animal to triangulate the location of the fish that it preys on in murky water. If you have a pet lizard, Many of your lizards might have a little spot on top of their head, which is sometimes referred to as its third eye. It is a light, sensitive patch of skin that does not give them visual capability, but it allows them to distinguish light from shadow. And that helps the lizard to know, for example, when it's out in the open, if a predatory bird is flying overhead and it needs to take cover. So there are certainly senses that exist in the animal kingdom that we do not have. It is altogether possible that there are senses that exist in the animal kingdom that we don't even know exist because we lack the ability to detect that kind of information that some animal might be considering as part of its day-to-day experience. For example, it has been observed frequently throughout history that there is often very strange behavior among rats and other rodents right before an earthquake, that in the hours prior to an earthquake, rats will begin running around, panicking, acting very, very strangely. And it's thought that they may have a vibrational sense that we lack, which makes them sensitive to deep vibrations far down in the earth that we are completely unaware of, that tell them in advance that something bad is about to happen. So, how many senses do you have? It's not an easy question. There's no actual agreement on that. Now I know what you're thinking. Back in elementary school, they taught you had 5 senses, right? Vision, hearing, smell, taste, and touch. It's one of the first things you learn in your science classes when you're a little kid. Well, here's the thing about that. We left out a few, didn't we? We left out, among other things, the sense of equilibrium, because it's very hard to explain to first graders that your sense of balance is actually a sense. We left out the very peculiar sense of proprioception, which is your body's own awareness of where its parts are. Your sense of proprioception allows you to know that your hand is over your head when your eyes are closed, because you can feel where your own arm is. We don't really think about that. We say, of course I know where my own arm is. How do you know that? It's because you have a sense that tells you that. You have a specific sense that is dedicated to telling you where your body parts are relative to one another at a given moment, and that is how you know where your legs are. That is how you can feel when you move your arm. And the thing we call touch is not one sense either. The thing we call touch is a whole bunch of different senses. A thing you need to know is that there are no all-purpose sensory receptors. Any given receptor is only sensitive to certain kinds of data, and it won't be stimulated by other things. Your eyes can't hear. Your nose cannot see. Those kinds of receptors are not stimulated by that form of data. So any given receptor picks up a particular kind of stimulus, and only that kind of stimulus. And the thing that we call our sense of touch is an umbrella term for a whole lot of different senses, including contact, pressure. Actually, deep pressure and light pressure are 2 completely different senses. Texture, feeling the edges of objects, that is a distinct sense. Heat and cold are distinct senses. Feeling stretch is a distinct sense. Feeling vibration is a distinct sense. All of these things are things that you have separate, independent sensory receptors for. The question of how many exact senses you have, not to say anything about, say, the chemical senses within your body. Those are senses too. Your sense of how much carbon dioxide is circulating in your blood, that is a distinct chemical sense. Likewise, your sense of how much oxygen, or how much sodium, or your blood pressure, or the level of stretch in your stomach when you fill it up with food. All of those things are distinct senses with their own kinds of receptors. There is no exact agreement on exactly how many senses we have, because we believe that certain kinds of data are picked up by the same receptors. But the general consensus is that human beings have somewhere between 17 and 26 distinct senses. Now we can divide sensory receptors into a couple of broad categories. First off, any receptor that tells you about your body's internal environment is an interoceptor, I-N-T-E-R-O-C-E-P-T-O-R. Any sensor that tells you about the external environment is an exteroceptor, E-X-T-E-R-O-ceptor. You've got your proprioceptors, P-R-O-P-R-I-O O-C-E-P-T-O-R, proprioceptors. Those are the receptors for the sense of proprioception that I told you about, scattered throughout your joints and your muscles and so forth, to make you aware of your body's own position and the movements of its parts. We have photoreceptors, P-H-O-T-O, photo means light. Photoreceptors are light-sensitive organs, which would include things like your eyes and that spot on the top of the lizard's head that I mentioned to you before. We have chemoreceptors. Chemoreceptors are sensitive to chemical stimulus. So our sense of taste, which is technically called gustation, G-U-S-T-A-T-I-O-N, is a chemoreceptive sense. So is your sense of smell, which is olfaction, O-L-F-A-C-T-I-O-N. Your various senses for things like pH and oxygen saturation are also chemoreceptive senses. We have baroreceptors, B-A-R-O. Baroreceptors are sensitive to pressure. They make you aware of pressure within your blood vessels, pressure within your stomach as it fills up with food. We are even pressure on your skin from something pressing on it from the outside. We have mechanoreceptors. Mechanoreceptors are a broad range of receptors sensitive to mechanical stimulus. These would include receptors that detect stretch, vibration, edges, textures, things like that. We have and by the way, your sense of hearing. Hearing is a mechanoreceptive sense. It's technically the sensitivity to vibration in the inner ear. Vibration of sound is what you're hearing. And your sense of equilibrium, or your sense of balance, is also a mechanoreceptive sense. It's actually sensitivity to changes in the movement of fluid within channels inside your inner ear that allow you to determine which way your head is oriented relative to other things and which way your body is moving relative to the environment. We have thermoreceptors. Thermoreceptors tell you about temperature. We have separate receptors for heat and cold, defined for this purpose as warmer than my body temperature or cooler than my body temperature. And we have nociceptors. N-O-C-I-C-E-P-T-O-R. Nociceptors are the receptors dedicated to a special and specific sense known as pain. Pain is its own sense. Now, your eyes might hurt when I shine a bright light into them, but that's not your sense of vision responding. Your ears might hurt from a really loud noise going off near your head. That's still not your hearing responding. Pain is its own sense with its own receptors. And it's an extremely important adaptive sense. The sense of pain is the one that tells your body when something is wrong. Nociceptors detect noxious stimuli. They warn you about tissue damage, about discomfort from toxins, discomfort from infections. There are a wide range of things that nociceptors tell us about, but they are the body's internal warning system that says something unpleasant is happening to me, you should do something to change that situation. So we may not like pain when it's happening. Well, okay, maybe you do. I'm not going to get into your personal life. But my point is that pain is an incredibly important adaptive sense, and in the absence of it, you would be in big trouble as an organism because you would not have an internal warning system letting you know when something was wrong with your body. This is a sampling of the kind of senses that we have and the broad categories of sensory receptors that carry them out. There are kinds that I haven't mentioned. Birds have magnetoreceptors that you do not have, for example. Not all animals have all of these senses, and not all animals are equally good at them. More on that momentarily. Now, any given sensory receptor, when it is stimulated by that particular kind of stimulus, whenever it gets a light shining on it for a photoreceptor, or whenever it feels pressure being applied to it for a baroreceptor, that will set off chemical reactions within the receptor that will send an electrical stimulus flowing down a nerve fiber. That nerve fiber will carry that stimulus to the central processors found in the brain and the spinal cord. Whenever a signal is flowing towards the central nervous system, we say it is going in the afferent direction, A-F-F-E-R-E-N-T, afferent. So afferent flow is flow towards something. So that's flowing in towards the central nervous system. Now, there are dedicated fibers for any given receptor. That is, your brain basically knows which nerves are sending in which data and interprets that data accordingly. We call that labeled line code. So basically, all this data coming in along your optic nerve, your brain says, okay, that's visual data, and it interprets it as light. Whereas all of the data coming in, for example, along the vestibulocochlear nerve that comes from your inner ear is being interpreted by your brain as either sound or as shifts in equilibrium, context determining which. If this gets messed up, your brain can misinterpret data. There is a condition some of you might have heard of called synesthesia, S-Y-N-E, or actually the proper spelling is S-Y-N-A-E, the digraph A-E often fused together as one single letter, S-T-H-E-S-I-A, synesthesia. Although sometimes you'll see the A dropped and just put it with an E these days in more modern spelling. But synesthesia is a failure of labeled line code in which your brain misinterprets data coming along a given pathway and says, okay, I'm going to interpret this data as the wrong sense. I'm going to hear smells, or I'm going to see flashes of color associated with music or whatever. Or I'm going to taste numbers, and when I read a number, I'll find the number 7 tastes bitter or whatever. Different people with synesthesia, it's not one condition, depends on which way your brain is mixing up the labeled line code, but different synesthetes will experience different sensory phenomena in different ways. My partner will get color associated with music, and when she hears a song, she'll say, oh, I like that song, it's blue, you know, that kind of thing. So it's not one condition, it's an umbrella condition. We don't have a good reason why synesthesia happens. It has not been well studied, but for reasons that should probably be quite clear, a lot of artists, musicians, and other people involved in the arts have reported synesthesia, and of course it can be temporarily induced in just about anyone with certain psychoactive drugs. All right, so one last thing about all this. You may have the same senses as your dog, but you are not equally good at them. Your dog has way crappier vision than you do. Dogs are not as good at seeing things distinctly. Their vision is not as sharp as ours, nor do they see the range of colors that we do. Your dog has a far more powerful sense of smell and hearing than you do. Different animals prioritize different senses based on their necessary environmental adaptations. Primates, in general, have prioritized the sense of vision. Vision is an extremely helpful sense for navigating the treetop environment in which primates evolved, seeing where branches are and how far away they are so I can climb or leap to them, gauging whether or not they are thick enough to bear my weight, noticing fruit tucked among the leaves. That's one reason primates have color vision, so we can find food more effectively in the treetops. And so we've adapted the sense of vision as being our most effective sense. Your dog, remember, is a wolf. Wolves are pack hunters and carnivores. They benefit from a sense of smell to help track down their prey and a sense of hearing to notice the sounds of it. So those are the senses they have prioritized. They're nocturnal hunters. There's less light available. Vision isn't as important for them, and therefore they are best suited to navigate using their noses and their ears. So different animals will have different sensory acuities depending on the particular adaptations that their environments demand of them and the things that they need to be doing in that environment. So you might have one sense that is very much stronger than others. This will be reflected in the structure of your brain, that you'll have more brain space dedicated to processing relatively important senses. The olfactory bulbs of your brain, B-U-L-B-S, are pretty small compared to those of a dog or even a sheep. Whereas the occipital lobe of your brain, which is the primary visual processing center, takes up almost the entire back of your brain. We dedicated a lot of brain space to it. So brain structure evolves basically to help interpret sensory data. By the way, there's a common belief, you've probably heard this before, that if you lose a sense, your other senses will become stronger to compensate. That if you lose your vision, for example, your sense of hearing will get stronger. That is not true in any way. What is true is that you will be paying more attention to that data, and your brain will be able to allocate space for processing it that was previously taken up by another sense. So your sense of hearing does not get stronger if you go blind. Your ears have the exact same ability to detect sounds that they had previously. What has happened is because you're not taking in visual data, you are paying way more attention to your sound data, and you have brain space that was previously dedicated to processing the complexities of vision, which can be reallocated to process the details of hearing. So you're getting more information out of the same received data. You're paying more attention, and you're able to extract more processing power from the brain in order to analyze what it tells you. All right, so that's sensory input. Sensory input, receptors are stimulated by the thing they are sensitive to, signals flow towards the central nervous system in the afferent direction. That brings us to step 2. By the way, we're talking about sensory neurons. If you're sensory neurons, those are neurons that are specialized to carry sensory data. Neurons, N-E-U-R-O-N-S, those are nerve cells. We'll talk about them more in a few. Next step is integration, also called information processing. Integration or information processing is when your brain takes the data that has been gathered by your senses and determines what, if anything, needs to be done about it. During the information processing stage, different parts of your central nervous system are engaging with different kinds of information. You probably are aware that you have brain centers, for example, that are dedicated to processing speech, and other brain centers that are dedicated to processing tactile sensation from your skin, and so forth. More than one part of your brain is going to be involved in processing any given piece of data. You right now are listening to my lesson. You've got parts of your brain that are interpreting the noises I am making in order to sort out the meaning of the speech. You have other parts of your brain that are making connections between the noises I am making and what those noises mean. Saying, okay, the word chair means a particular object. When you say the word chair, a mental picture of a chair probably pops up in your head. That's another center of the brain telling you this is what a chair looks like. You've got other centers of your brain that are going to hear me say the word chair and are going to think about the chair that you're sitting in while you listen to this lesson. Or they're going to remind you, oh, I need to go shove something under the leg of that chair that's wobbly, or what have you. Lots of different centers of your brain get engaged to process any one piece of information. It's not all happening in one single spot. That means that if you have problems with one particular part of your brain, you may have a situation in which you can do part of processing, but you can't do all of processing. There is a condition called aphasia, A-P-H-A-S-I-A, that is the inability to speak. Or more to the point, aphasia is the inability to speak properly, basically because although you can understand speech coming in and you know the words you want to say, the part of your brain that is making the connection between words I know I want to say and muscular actions involving my vocal cords, lips, mouth, and tongue to form those noises, that connection is being broken for some reason. And as a result, you're not able to say the words that are in your head. You can understand when somebody else says them, you can't make yourself say them. Similar condition called prosopagnosia. Prosopagnosia is a condition in which you are unable to recognize faces. You can see people's faces, but you cannot make the link mentally, at least not easily, between that human being's face and saying, oh, that is Tyler. You know, that's you can see his face just fine. You can see all of his features. You can look at Tyler, but you don't remember that that is the face of Tyler. I have a friend who has this condition, and when I'm talking to him, if I see him, I say, hi, it's Adam, because I know he's not going to know who I am looking at my face. All right, so during integration, different parts of your brain are doing different aspects of the processing, collating all the different forms of data. Right now, when you're watching my lecture, you've got a bunch of different kinds of data coming in. Visual data from looking at the images on the screen, auditory data from the noises I am making, and you've got several centers of your brain engaged to put those data together, to integrate them into one comprehensive picture of what is going on, to interpret what is happening, and to apply previous experience to it. That's a big important part of this. Integration involves accessing memory and saying, what do I know about this stimulus? What am I aware of about it? Based on previous experience, do I know what that noise means? Can I interpret what that sight is that I am seeing? Do I know the implications of the data that is coming in? That is all part of the integration process. A vitally important part of integration is also forgetting. It is discarding irrelevant data and saying that data right there is not important to the thing I am doing right now. What matters at the moment is listening to Mr. Davis's words telling me how my brain works. I don't need to devote brain space to thinking about the color of his sweater or the texture of my socks. Not important right now. Except now they're important because I just drew your attention to them, and your brain says, oh, I obviously need that for some reason. So this is all involved in integration. There's a tremendous amount of sophistication involved here in taking all that data that came in, discarding the irrelevant data, putting together all of the other data that just arrived, gathering its meaning, and ultimately determining what, if anything, you need to do about this. Making decisions about, does this data demand a particular response from me? I could say a lot more about integration. The whole cognitive process is an incredibly fascinating one, and frankly quite far beyond the scope of this class. If you take an anatomy and physiology class or a neuroscience class, you'll hear way more about it in there. And finally, we have the last step. The last step is motor output. So integration happened in the central nervous system, the brain and or nerve cords within the body. I say and or because your spinal cord actually makes certain decisions for you. Your spinal cord handles most of the data processing involving basic reflex actions like defecation, urination, and orgasm. The conscious brain is not involved in a lot of that information processing. You can step in and say, okay, I don't want to pee right now, but that's about the extent to which your conscious mind handles that processing. Your spine handles most of it. Okay. By the way, different kinds of material you will see in the brain and spinal cord. You've all heard of gray matter, right? Gray matter versus white matter. If you look at a brain, the outside surface of it will be a nice grayish color. That is the gray matter. The gray matter is the processing parts of the brain. Cut down into that brain and you will see the white matter, which actually makes up the bulk of the brain. The gray matter forms what we call the cortex, C-O-R-T-E-X, on the outside of the brain. There are lumps of gray matter deeper inside the brain called the nuclei, but most of your brain is white matter. White matter is the communication system. It is basically the pathways, the bundles of nerve fibers that connect one part of the brain to another and allow the brain to talk to itself. The white matter is organized into bundles called tracts, T-R-A-C-T-S. So that's gray matter versus white matter. Gray matter does the actual processing part. It consists of what we call interneurons, which integrate information. White matter broadcasts data from one part of the brain to another, allowing it to make connections between information. An important part of brain development, incidentally, is forming connections between different centers of the brain and also in pruning away connections that are not as useful. It is believed that the process of synaptic pruning, which happens a lot when you're a kid, is a big determinant in how you think later in life. Your brain forms the bulk of its connections when you're quite young, which is why exposing kids to a wide variety of different kinds of stimuli, different sounds, different sights, and so forth when they're very young will give them greater cognitive capabilities later. There's a thing that went around a number of years ago called Baby Mozart, in which they said play Mozart music for your kids when they're little, because the complex melodies of Mozart music will form more complex connections in their brains and help them to do more kinds of things as they get older. We don't know how true that specifically is, but there definitely seems to be evidence backing up that more complex stimuli that kids are exposed to when they're young will help them form a wider range of synaptic connections within their brains. By contrast, a thing called synaptic pruning, P-R-U-N-I-N-G, and synaptic, S-Y-N-A-P-T-I-C. Synaptic pruning is the process of basically removing non-useful connections, basically removing places where two regions of the brain linked up with each other in a way that they should not have. It is believed that a failure of synaptic pruning, that a place where the brain did not break unhelpful connections when you were young, may be responsible for autism spectrum disorders. And that's still being explored right now. All right, then. But ultimately, your brain has taken in some data. It has processed that data and determined what to do about it. Let's assume, for the sake of argument, that it's decided you need to take some kind of action. Now we get to motor outputs. Motor output is when we send instructions from the central nervous system out to the body. Motor outputs is going to be carried out by motor neurons, as you can probably guess. That's M-O-T-O-R, by the way, not E-R. Motor output is an electrical signal leaving the central nervous system, traveling along nerve fibers that are attached to the brain or to the spinal cord. We have cranial nerves, the ones that come from the brain, C-R-A-N-I-A-L, and we have spinal nerves, the ones that come from the nerve cord. Those nerve fibers then travel to whatever body structures can carry out those instructions. We call any structure that responds to that signal an effector, E-F-F-E-C-T-O-R. Your body has two primary kinds of effectors, muscles and glands. Almost everything your body does can be facilitated either by moving a muscle, whether it's a consciously controlled muscle like your biceps in your arm, or whether it's an unconsciously controlled muscle like the smooth muscle in the walls of your stomach to churn your food. Or it can be facilitated by secreting something from a gland. You need to adjust your blood pressure, secrete the hormone aldosterone, which is one of the hormones that controls that. You need to start digesting your food. Let's squeeze some glands that secrete gastric juices into your stomach. So a lot of our basic actions are really mediated by either a muscle doing something or a gland doing something. So those effectors get the signals from the brain, which stimulate them and tell them what to do. We're not going to go into the complexities of how exactly a muscle responds to a brain signal or how a gland responds to a brain signal, not in here. Take a neurology class for that or an A&P class, but this gives you the basic pathway. So that is our fundamental description of how nervous systems respond. Sensory data comes in the afferent direction along sensory neurons. It is processed by the central nervous system using interneurons, and then instructions go out in the efferent direction, E-F-F-E-R-E-N-T. Afferent means coming in, efferent means going out. So motor output goes in the efferent direction towards effectors that can carry out the brain's commands, whether you're consciously aware that effector is doing so or not. And your brain, by the way, does not inform you about a lot of the things that it's doing. It doesn't need to, because a lot of that stuff happens in the background. Your brain does not have to tell you, okay, I'm secreting gastric juice now. You don't need a sensation from that. Food arrived in your stomach, stomach begins secreting the stuff needed to break it down. All right, so that's our broad-based picture of nervous system functionality, and that basic story I just told you is pretty much the same in every animal with a central nervous system that is able to process data. Now, the actors in all of this, the main active cells in nervous tissue are cells called neurons. Neurons are rather extraordinary cells. They are the longest lived cells in your body. You still have neurons you had when you were a baby, and you will have them until you die. Neurons are rather remarkable. They are highly sensitive to electrical stimuli and respond very, very precisely to it. They're much more excitable and conductive even than muscular tissue, which also has those properties. Now, neurons are among the longest cells in your body. And here is a picture of a neuron. Can you see that? You cannot see that. There we go. Okay. Here is a picture of a neuron. That bluish mass you can see on one side, that is the cell body of the neuron. That is sometimes called the soma, S-O-M-A. It's going to contain the cytoplasm, the nucleus, all the different organelles that you would naturally expect to find inside any kind of eukaryotic cell. You can see a nucleus there. One thing that neurons notably do not have is they do not have centrioles, which you might recall are the organelles involved in cell division. Neurons cannot divide. And that's why if you get a nerve cut through, you're never going to heal it. That's why if you get your spinal cord severed, you can't walk again, because your body does not build new neurons because the cells are not divisible. Now, extending from the cell body are a number of branches or processes. A process, when you use that term anatomically, means a branch sticking out from something. You can see all around the soma, or the cell body, we've got a bunch of branch-like processes, kind of tree-like processes, called dendrites, D-E-N-D-R-I-T-E. Dendrite, the word dendrite means little tree. You can see why. Dendrites are highly branched processes. Their job is to bring data into the neuron. They are the afferent processes. Data comes into the neuron through them. Whenever a sensory receptor is stimulated, that receptor sends its signal into the neuron via the dendrites. The more dendrites a neuron has, the more data it can take into account. Now, extending away from the neuron is a single long process called the axon, A-X-O-N. The axon is the efferent process of the neuron. It conducts signals away from the neuron's cell body. The axon can be up to 2 meters long. At its far end, the axon branches into what we call the axon terminals, these long branches. The axon terminals branch extensively. At the ends, they have what we call terminal buttons. The terminal buttons are going to connect to either the next neuron in line, or they're going to connect to an effector, that is a muscle or a gland. The connection point between a neuron and its effector is called a synapse, S-Y-N-A-P-S-E. That word is both a noun and a verb. I can say here is the synapse connecting this nerve to this muscle, or I can say that nerve synapses with that muscle. Now, the axon is protected by a protective barrier of cells wrapped around it, forming what we call the myelin sheath, M-Y-E-L-I-N sheath. The myelin sheath, or the myelination, acts exactly like insulation around an electrical wire. It prevents the electrical signal of the neuron from grounding out before it gets to where it needs to go. The myelin sheath is made up of Schwann cells, capital S-C-H-W-A-N-N, named after Theodor Schwann, the German anatomist who first described them, and also one of the fathers of cell theory, incidentally. So the Schwann cells form the myelin sheath. They wrap around the axon to insulate it and conduct the signal to where it needs to go. There are small spots where the myelin sheath does not cover all of the axon. Those are called nodes of Ranvier, N-O-D-E. Ranvier, capital R-A-N-V-I-E-R, after the scientist who first described them. Should be Ranvier, but of course we never pronounce it right. So say Ranvier or nobody will know what you're talking about. Sometimes a little side branch of the axon shoots off from a node of Ranvier. If that happens, we call that an axon collateral, C-O-L-L-A-T-E-R-A-L. But that is unusual. Typically, it's this long, single, straight process until you get to the branching axon terminal at the end. So my neurons are the active cells of the nervous system. They receive data coming in via their dendrites. That sets off an electrical stimulus, which is conducted away from the cell body along the axon. Then it travels along the axon, gets to the axon terminals, where a synapse between the axon terminal and the next cell in line, whether it's an effector cell or another neuron, passes that signal from one to the other. Okay, so those are your neurons. They are the active cells of the nervous system. They're the ones that are actually sending the signals out. What I just showed you is a fairly typical version of what is called a multipolar neuron, meaning that it has multiple axons and one axon, multiple dendrites. Some neurons are going to be bipolar. They have one dendrite and one axon. Some neurons are going to be unipolar. They have one single fused fiber that's both dendrite and axon. But multipolar neurons are far and away the most common ones that you see. So that is going to be the basic structure of our nerve cells and the things that they use to send signals. Now, at the end of the neuron, the signal is going to arrive at the terminal button. Within the terminal button, you're going to have vesicles full of chemicals called neurotransmitters, N-E-U-R-O-T-R-A-N-S-M-I-T-T-E-R, neurotransmitters. Neurotransmitters are the chemicals used by the nervous system to send signals. Now, I didn't tell you a thing before, but when the neuron arrives at the next place in line, the synapse is not actually a connection. between the neuron and the effector. The synapse is a little enclosed capsule that the neurotransmitters will travel through. The synapse is a gap. There's actually a little gap, a space between the end of the neuron and the cells of the effector. We call that the synaptic cleft, C-L-E-F-T. A cleft is a gap or a groove. So the neuron doesn't actually touch the muscle fiber or the gland or whatever is next in line. It just comes almost all the way to it, and then the synapse is a little enclosed, surrounded by a connective tissue capsule around a little gap between the neuron and the next cell, only a couple of nanometers wide. Whenever the electrical signal arrives at the end of the neuron, it stimulates exocytosis, whereby the vesicles move to the edge of the membrane and release the neurotransmitters. The neurotransmitters then travel across the synaptic cleft, bind to receptors on the far side of the cleft, and that stimulates the signal to continue on the other side, either carrying the signal further along or stimulating the effector to act. And that's the basic way that our synapses work. Now, there are different kinds of neurotransmitters. You're probably all aware a little bit of neurotransmitters. There are different sorts of them that do different jobs. Some of the more well-known neurotransmitters in animals, and these are almost universal, are acetylcholine, A-C-E-T-Y-L-C-H-O-L-I-N-E, often abbreviated capital A, capital C, little h. Acetylcholine is the primary neurotransmitter of the skeletal muscles. It's the one that stimulates skeletal muscle to act. Incidentally, acetylcholine binds to the same receptors as nicotine, which is why smoking often causes a bit of jittery nervous agitation. We're going to find that we have epinephrine and norepinephrine. You've probably heard of those. E-P-I-N-E-P-H-R-I-N-E, that's epinephrine. Norepinephrine is N-O-R, epinephrine. Those are collectively known as your adrenaline hormones. Those are neurotransmitters that stimulate the fight or flight response in which your body gets ready for action. More about that later. We have a neurotransmitter called dopamine, D-O-P-A-M-I-N-E. Dopamine is the pleasure sensation. It's released whenever your body experiences pleasant stimuli. Dopamine is very important to animals because it is responsible for what we call reward learning behavior, whereby an animal keeps doing a thing because that thing feels good. And this is a really important adaptive response because you eat some tasty food and it tastes good, right? That good feeling is because of the release of dopamine. Dopamine makes you feel good. And so that tells your brain, ooh, this is good stuff, I should keep eating that. And that's how your brain learns which foods give you the nutrients your body requires. Likewise, sex feels good. That's because you get a reward of dopamine for that. And that tells your body, hey, this is a good thing to do. Let's keep doing that. Whereby that means your DNA is doing its fundamental imperative, being passed on to potentially reproduce. So dopamine is vitally important to animal adaptive response in learning which behaviors are biologically quote-unquote good for you, if you are. Even if it's not good for you in the sense of eating too much of the sugar is bad or whatever, but it is good for you in the sense of fulfilling biological imperatives. Dopamine is also very involved in regulating pleasure, sleep, mood, a lot of aspects of your behavior. You probably are aware of serotonin, S-E-R-O-T-O-N-I-N. It is your body's primary mood regulator. Regulates anger, aggression, libido, mood, appetite, body temperature. All these things are tied to serotonin. It's a very important metabolic regulator. Imbalances in serotonin levels are responsible for a significant number of psychiatric disorders. I'm sure many of you are aware that imbalances in serotonin, too much or too little, can be responsible for such conditions as clinical depression, bipolar disorder, psychoses of various sorts, schizophrenia. All of these things can happen because of basically dysfunctional serotonin levels. We have a bad tendency in our society to believe that mental illness is sort of your own problem, that you just need to get over yourself and, kick it yourself. That's a very, frankly, hostile and non-compassionate way of looking at mental illness, grounded in a very, very old set of beliefs that you probably don't consciously hold anymore, but for a long time it was believed that if you were mentally ill, it was either because you were sinful and weak of character or because you were possessed by a demon, which could only happen if you were already sinful. In other words, the general belief for centuries was that if you were mentally ill, it was because you were a weak, bad person, and it was therefore basically your own fault. And that led to not a lot of sympathy for mental illness. We don't like to believe that a lot of our behavior is hard-coded. We do not like to think of ourselves as meat machines whose personalities can be modified by structural and chemical damage. We like to think that there's an immutable me inside of there somewhere, a self that is basically carried around in this meat sack and is independent of anything happening to it. That self has to be filtered through that meat sack. And that means that if something goes wrong with your neurotransmitters, your behavior and your personality will be affected. These are clinical features, and when somebody is mentally ill, frankly, you need to be compassionate about that and treat it as an illness, the same way you would treat them as if they were having a lung problem or a heart problem or a pancreas problem. You would tell a diabetic, just try to smile and get over yourself and the insulin will feel better. You wouldn't tell an asthmatic, you need to just buck up, and start breathing more properly. That's improper breathing behavior. No, you give them treatment. Give mental illness treatment. Don't stigmatize it, and certainly don't feel bad about having it or needing help for it. The sooner we get over that, the better off we're all going to be. And finally, thank you for coming to my TED Talk. And finally, endorphins. E-N-D-O-R-P-H-I-N-S. Endorphins are your body's natural analgesics. A-N-A-L-G-E-S-I-C-S. Analgesics are pain blockers. Your body produces natural pain-blocking chemicals because some of the things we do to our bodies hurt, especially when you're pushing them super hard. When you are exercising, when you are frightened, your body releases endorphins. Partly that's because the stimulus of exercise can be painful to muscles and joints and so forth, and that's helping to counteract that so you can keep doing what you're doing. Partly that is preparing you for the possibility of injury. That when you're scared or moving around forcefully, your brain is aware, hey, I'm doing things where I might get hurt. Therefore, releasing a flood of analgesics basically makes your body ready to deal with the forthcoming pain. Incidentally, heroin and morphine, our opioid drugs, binds to the same receptors as endorphins do, and they produce similar effects. Addiction to them is quite easy to come by. Addiction is also a disease. Anybody who tells you differently has not studied their brain chemistry. Some people are way more prone to addiction than other people are. Some people will get addicted on the first hit of morphine that they get after surgery. Some people will be heroin users for years and not get addicted. So don't blame addicts. They're in a bad situation. Help them out. It's not a good thing. But anyhow, though, your analgesics are interesting because basically they're getting your body ready for the possibility of pain and helping your body to deal with it when it's hurting. So whenever you are hurting, your body releases analgesics like endorphins, to basically try to dampen down the pain, which is why most pain will dampen down after a while. Fun fact about this, if you're not actually in danger or you're not actually suffering from injury, that flood of analgesics will make you feel on top of the world. The runner's high, some athletes call it. You've all probably felt it if you're an athlete, and you're going and suddenly you get in the zone and you break through the wall and you start feeling really, really good and you feel like you can do anything. Yeah, that's because you got way more endorphins than you need because there's not enough pain for them to counteract, and that means that you're just going to feel the good sensation from that. It's the reason why we, feel good whenever we're in the middle of a good workout. It's the reason why you feel good after you ride the roller coaster or go on the haunted house, because the fear stimuli, did I say go on the haunted house? Go through the haunted house. Because the fear stimuli flooded your body with endorphins in preparation for the possible danger, and now you feel good from that. It's why we like horror movies. It's why we like thrill rides. It's why some people like sadomasochistic sex, because they're getting a pain stimulus, but the endorphins flood in, and there are way more of them than the pain they're receiving. Okay, so that's kind of a quick look here at neurons and how they work. Now I see that we've been going for about an hour here, so I think I'm going to cut this short. We have a bit more to talk about to finish up animal nervous systems. And by the way, all those neurotransmitters I just described are pretty much the same across the animal kingdom. You will find that although some animals produce different ones than we do, or not the same ones at the same levels as we do, at least in vertebrates, everything I just described to you is pretty standard. Okay, so Next lesson, our final lesson, I think, on animal nervous systems, we're going to talk about the support cells of animal nervous systems. We're going to talk a little bit about the central nervous system in vertebrates, talk a little bit about the brain and its organization, and we're going to talk a little bit about the divisions of the nervous system, structural and functional. Sound good? Okay. Thank you for all coming today. Hope you're having a good afternoon. I'll talk to you soon.